So a quick introduction here for just a few minutes. And first and foremost, thank you to Mark Chasen for agreeing to give the presentation today. So a quick bit of background on HiFi for structural variant detection. So when we think of structural variants, there's all sorts of different types of variants out there. So there's things that are not structural variants. So we think of things like small single nucleotide variants or small indels. There's probably different definitions of how big those have to be, but one is being less than 50 base pairs. So, you know, single nucleotide variants, but then we can think of larger things. So things greater than 50 base pairs. So you can think like duplication events, whether those are tandem side by side versus interspersed at another location, inversions, translocations, copy numbers, or even things more specific like we'll talk today about tandem repeats and VNTRs. So why HiFi sequencing for structural variant analysis? Well, one is HiFi reads span these large events. So for instance, here you can see on the top some PacBio HiFi reads aligned, and you can see this clear deletion event. And then we see short reads aligned similarly, and you can see where the coverage doesn't go too great there at the repeat. So this deletion is not being detected. So there's different estimates about how much you can miss based on using shorter versus longer reads. Just as one example, here's a paper in Science using 32 samples from the 1,000 Genomes project, where we see about 9,000 variants being detected per genome with short reads, whereas with long reads, that's about 2.5 times the amount, so about 24,500 events per genome. And why do we care about picking those up? I mean, one way of thinking about that is in what type of variants we can detect when, say, analyzing a human genome. If you think back in the day, we used to use karyotyping, so for finding chromosomal abnormalities where we could, you know, explain about 5% of disease. That got better as time went on. We used microarrays where we could get copy number variants, maybe explaining about 10%. Then we had the advent of the exome and short read sequencing. So we did the explosion of, you know, going from 10% maybe to a 30% explanation rate, where we could detect single nucleotide variants, indels, and some large exonic variants. Going from exome to short read genomes, it gives us another jump in detection. So moving up to about 40%, because we could add in, you know, all those parts that were getting missed by the exome. Plus, you have a more even coverage, so you know maybe some of those events are easier to detect. But really, then estimates are just leaping when you look going from, like, say, a short read genome to a long read HiFi genome, where we have everything we were detecting before. We still have the single nucleotide variants, indels, and those larger variants that were detected. But on top of that, we're also now picking up these, you know, as we said, you know, maybe 2.5 times more structural variants than before, which can help explain maybe up to 67%. That also includes phasing of the variants, and as well as inversions, repeat expansions. As we mentioned, we'll talk about tandem repeats here in a bit. Some of the tools that we offer actually from PacBio itself is, let me see, PBSV. So PBSV is a tool for finding structural variants. It varies a bit to how it runs depending on what type of variant. So whether that's an insertion, a deletion, an inversion, a duplication, or a translocation. These are the optimal default settings for detection. And the way this is running is we're running an alignment, and then after that alignment, looking for those signatures for the structural variants, as well as clustering, support, and then basically make it a consensus. So maybe a lot of you in the audience have used PBSV, but maybe not many of you know that there's also this command line thing called SVPack, which is also basically on GitHub, so it's public, for filtering, comparing, and annotating those structural variant calls in VCF format. And also in the past year, there's been a new tool released by PacBio called the Tandem Repeat Genotyper, or short Target. Again, this is also available in GitHub. If you Google, you can also find on YouTube a presentation revealing the unseen in human genome sequencing. That'll give a lot more details. But just to give you an idea of how this is working is it uses as input the aligned HiFi reads, as well as definitions of these target repeats or target tandem repeats. So for instance, the position, as well as how that tandem repeat is defined in its structure. And from that, it can give you in the results both the repeat sequences and links. On top of it, we can do methylation calling. A big advent for PacBio last year was the native 5MC calling, so that's incorporated. And then what's also great is so often these things are hard to visualize in traditional genome browsers. So it also provides the Tandem repeat visualization tool, so TRVZ. So here you can also see that plot where we can see the flanks of this Tandem repeat, as well as the size and also insertions, mismatches, etc. So looking at where we are today, I mean, it's been an exciting couple months since the release, the announcement of the Revio system. I think this is really also changing things. What's really cool about Revio is basically one SMRT cell is about a 30x human genome. So that's helping us both in terms of scales and the number of samples, as well as the affordability. This is, of course, based on US dollars, a little under $1,000 in terms of sequencing reagents. Also on board, we've now moved from our internal consensus to deep consensus for calling, and it's also much easier to use. And just for a little bit of a comparison, here's the previous Sequel IIe system to the brand new Revio system in terms of its accuracy for single nucleotide variants, indels, and structural variants. And we see basically equal or even actually slight improvements in the calling. So it's getting easier and easier to do this type of analysis. So moving on, I first want to thank Dr. Chason for presenting today. He's, really been one of the pioneers for using long reads to detect structural variation. Going back to the days when he was at the Eichler lab doing, for instance, the hydidiform mole, which was, kind of the first genome where we really saw those big differences in what could be detected between long and short reads. So basically, here's two papers example, a 2015 Nature paper where he was first author, and also one 2 years later, all the way to on more recent papers. So moving from those single haploid genomes to not only diploid genomes, but diploid populations. So here's this paper that was also referenced earlier where we saw 2.5 times the detection of structural variants using long reads than short reads. And with that, I'll hand it over to Mark to present our keynote talk today.
Thanks, Matthew, for the introduction. I'm going to be talking about a couple new methods that we've developed for detecting variation in tandem repeat sequences. I know a lot of you are here to see a talk on long reads. Unfortunately, some of the data is still coming out for long read sequencing. I'm going to tell a two-part story, one where we're making some inferences on what we could potentially find with short read data. And then I'm going to talk about a tool that we've developed for kind of carefully annotating tandem repeat variation with long read data. The work that I'm going to talk about was done by 2 students and a postdoc, Jingwen Ren, Bita Gu, and Tony Liu. It's part of my lab down here at US. So we all know that long read sequencing of populations is growing now that we have faster machines to sequence more samples. And so we want to dive a little bit into so it's well known that given a long read data set, look at the frequency distribution of structural variants that we find and compare that to Illumina sequencing. On similar distribution for Illumina sequencing, we have this large gap between what's discovered with long read data versus short. This is a histogram from the science paper that Matt mentioned just a few minutes ago. And there's a few fold increase in the number of structural variants observed in long read data versus short read data. And this has been a well-established pattern for many years now. What's important though is if we look at the particular variants that we find in that data set and say, intersect them with the boundaries of tandem repeats on the human genome, we can see that about 2/3, so 66% of insertions and 60% of deletions are fully contained within tandem repeats. Okay. And to give kind of an overview of tandem repeat sequences in the human genome, these are tandemly ordered repeat arrays that have motifs on at least 7 bases. So between 2 and 6 are short tandem repeats. They average about 215 bases. And what's interesting is they aren't perfect repetitions of motifs. They have differences in motif composition in addition to length. And interestingly, they tend to be underrepresented in the human genome. So this is a disease-relevant tandem repeat that we like to study in this lab called the CACNA1C intronic tandem repeat. And this is a dot plot of the tandem repeat against itself, and also the similar tandem repeat in the human genome, where much of the sequence is actually missing from the HE38 reference. But what you can see here is that the particular pattern changes over the various location in the tandem repeat, based on the sort of different orthogonal patterns that you can see here. So when we first started noticing that there's a lot of these new structural variants that we find in long-read sequencing are in tandem repeats, we're asking, okay, what's the global diversity? So before, when we only had a few samples, we didn't know what these look like across populations. And what's the functional impact of tandem repeat variation? And the unfortunate part about asking that question is that current large-scale diversity and functional data is only in short-read data sets. Diversity data is changing, hopefully pretty rapidly, but the functional data is still kind of lagging. And so one thing to kind of remind ourselves is that in unique regions of the genome, short-read data actually maps pretty well. And here is kind of a little example of actually an allo insertion that's absolutely obvious in long-read data. It's actually pretty easy to detect in short-read data. We can find this type of variation moderately well, and we have a good idea of its impact on diversity and disease. But when we go to tandem repeat variation, this is a browser view where the light-colored bars represent low-quality, low-confidence alignments. We can see that there's either no coverage or low-quality coverage with the short-read data in the tandem repeat sequences. This makes it very difficult to detect variation in these types of regions when you only have short read. So the first part of the story is a quick kind of overview of a method that we have using pan-genomes on repeat graphs to be able to detect variation using short read data in tandem repeat sequences. So our solution for doing this is we essentially build a pan-genome that reflects the repeat structure of. So that's the pan-genome that we build is actually a very kind of aggrandized name for what's just simply a De Bruijn graph that we build on tandem repeat sequences, given a bunch of assemblies of various human genomes, say from the Human Pangenome Project or the Human Genome Structural Variation Consortium. So the reason why we use this De Bruijn approach is that when you have a repeated sequence, it shows up as kind of a tidy cycle in the De Bruijn graph that easily represents the polymorphic repeat natures in terms of paths that just simply traverse these cycles multiple times if the repeat is in different copies in different individuals. And then simply, if we want to make this a pan-genome graph and we add additional sequences, this just corresponds to adding extra vertices and edges in the graph that add additional cycles for read analysis. And so my former student, now postdoc, Tony Liu, wrote a method called, or a toolkit called Danbing TK that essentially takes a set of regions of tandem repeats in the human reference as well as a few assemblies, and essentially collects all of the VNTR sequences independently from the reference and all assemblies, builds individual pangenome graphs, or essentially De Bruijn graphs, and then is able to map reads into these graphs. And what that looks like is we essentially, when we map reads, we essentially count how often reads align to individual vertices in this graph. And what we can do is we can actually just count for some read data set how often reads align into the vertices that sum those up, and those give us an estimate for the total length of tandem repeat. And if we do something where, say, we map the 1,000 genomes data into this and look at individual loci, we can actually find stable differences in tandem repeats between populations. can actually find a few thousand loci where we can identify differential length between populations. So we can at least know that this approach, while it's fairly noisy because we're using short read alignments and essentially a read depth based approach, it can give us stable differences or biological differences between individuals. So the next step that we did was to use this approach to map EQTLs in tandem repeat sequences. And so I'm just showing the result from our more recent analysis, where our pan-genome, the high-quality haplotype resolved assemblies made by the Human Genome Structure Consortium, and we're able to find 2,768 cis EQTL, where the length of a VNTR that we estimate based on this read depth approach in the mapping reads to the repeat pan-genome graph is associated with nearby gene expression. And so we'll kind of dive into one pretty obvious example that has a bit of a twist. So the HRNR gene is a really a gene with no more than 2 exons, but one of the exon is actually this fairly long tandem repeat that in individuals ranges between 5 and 30 kb. So this is an obvious EQTL because you have a longer exon, therefore you're going to generate more RNA-seq reads that map back to this particular location, or that map back to this exon or the graph that corresponds to that exon. And what's shown here are the different repeat patterns of this individual gene. What's interesting is if we kind of zoom out, we can find nearby genes, flag AS1 and flag, that actually have a negative correlation of expression with respect to the length of this HRNR exon. And so this means that, okay, well, there's this obvious EQTL where you have a longer sequence, but that longer sequence is actually affecting the expression of other nearby genes. And if we look in a little bit more detail, what we can see is there's actually a topological domain kind of centered upon this HRNR gene. And if we look, and so we know that topological domains are influenced by the binding of the CTCF protein that can form to loop formation and changes in regulation. And so if we look at this HRNR sequence in a set of high quality haplotype resolved assemblies, we can actually see that there's a CTCF binding site within the exon. And the number of binding sites here increases as the length of the exon increases. One of the grad students in our department has done some research, some estimates for whether or not this is affecting the 3D structure. We just had it finished yesterday. We want to kind of polish it up before we kind of release it to the world. And I didn't get her okay on whether or not I could present this, her results. So I'm holding this off, but we think that the increase in length of this HRNR is affecting the 3D structure of the genome in this region. And we can kind of estimate that based on the number of CTCF binding sites in this particular exon. So I mentioned earlier that tandem repeat sequences vary both by length, where we can have, say, a reference that has an increase in, or a sequence that has an increase in copy number of a particular motif. They also vary by motif composition. So we can have slight differences between motifs and in addition to differences in copy numbers of motifs. And in fact, just like before, when we looked for stable differences in overall copy number, we can find stable differences in how different populations map to the different areas of the repeat. Oops, sorry. How different populations map to the repeat pan-genome graph. So this led us to ask whether or not it's possible to actually map, do kind of a finer grain mapping of EQTLs, where we look to see not whether the overall length maps, but individual edges or how reads map to individual edges if they're correlated with nearby expression. And we actually were able to show that there's actually quite a bit of cis-eQTLs that can be discovered when we test for association of individual motif counts with expression. So we're able to find over 25,000 motifs that are associated with nearby cis gene expression. And one kind of interesting example, like we mentioned before, this CACNA1C VNTR, it's a VNTR that is intronic to CACNA1C and was found a few years ago by Stanford researchers in David Kingsley's lab that the VNTR is associated with schizophrenia not by length, but by its composition. And we're able to show that there's actually a few motifs in this tandem repeat that are somewhat loosely, but they are associated with a decrease in expression of the gene CACNA1C. And we're able to show that additional motifs from what were discovered in the Song paper that were identified as risk alleles for schizophrenia that are also identified as risk alleles for a decrease in expression. So the 25,000 cis-eQTLs represents a large number of loci that are just in LD with other causal SNPs. And so what we're able to find, actually, if we run fine mapping, is that a much smaller fraction, but a positive or a non-zero number. So about 165 E genes are fine mapped to motifs with this PIP value then at least. We think that the motif composition is has a causal effect on nearby gene expression for a limited subset of tandem repeat sequences. Okay, so so we just talked about some kind of as quick as possible some results for discussing the functional impact of tandem repeat variation. What I didn't talk about was the fact that it took an enormous amount of effort to be able to get clean enough results to get these counts that we have. And we don't want to be able to have to do that going into the future. We certainly want to be able to analyze some of these large data sets, such as GTEx and UK Biobank that are all short reads. And great, we have methods to analyze those. But in reality, we want to be able to move to a world where we can get digital counts of motifs, not necessarily these estimates based on depth. And that's something that's starting to change with long read sequencing. And so last year, Doruk Beider published a paper in Nature Genetics looking at long read sequencing of Icelandic individuals. And there he was able to show that the copy number of tandem repeat in ACAN is associated with height in their cohort. And importantly, he was able to develop some particular methods for annotating the specific copy number of repeats in this sequence. And so we're able to get an exact count here. If we turn back to that science paper where we studied structural variation in 32 genomes or 64 haplotype resolved assemblies, where it had this statistic where there was this large number of structural variants in tandem repeat sequences, when we zoom in on the actual alignments, we can see this huge diversity between individuals. So here's just kind of one, not quite randomly chosen, but a known to be diverse tandem repeat, where I pulled 11 haplotypes, and there are 8 different lengths of tandem repeat insertion here. And unfortunately, a lot of the methods that essentially, or when you're sequencing a population of individuals, you want to be able to count, say, the allelic, the frequency of a variant in a population. And so you need to be able to merge variants across assemblies. So typically what is done in the variant merging is to find variants that are of similar length and similar position and essentially count them as the same. And in this particular instance, we have these 11 different sizes that are shown in, 8 different sizes and 11 assemblies, but there's only one variant that's actually chosen to represent all this diversity. And then not only that, that single variant is just represented by a length increase. So it doesn't have any of the extra information for the motif changes that are potentially happening here and are potentially having functional impact. So what we want is, like I said before, instead of just looking at the length, we want to be able to annotate the number of specific motif representations. Dr. Beider's figure from his Nature Genetics paper. When there's sort of subtle differences between motifs, we want to be able to identify the full composition. Okay, so we want the number of motif repetitions and the identity of the different motifs that are present in it. Okay, so what do we need to be able to do this? To be able to at least develop a method to do this. We need the coordinates of known tandem repeat loci in at least HG38, the human reference, or in a set of assemblies that we can annotate for tandem repeats. We need a collection of the different motifs that are observed in a population that we can use to annotate new sequences with a standardized annotation. And then we need a method for annotating motif composition, given new VNTR loci and sets of known motifs. So the tandem repeat coordinates, we can just grab those from HG38. There's a bit of a difference. Anyone sort of buyer beware, if you just download from the Santa Cruz browser, there's a lot of ALU sequences that are contained within the normal tandem repeat. You can choose to filter those out if you want. And then the next step is how do we get different motifs observed in the population at a locus? So our approach is to essentially take these diversity genomes that have been sequenced by either the Human Genome Structural Variation Consortium, again, 64 haplotypes, or the Human Pangenome Reference Consortium, which is having a rapidly growing number of assemblies. It should be at about 150 in the relatively near future. And we take the observed VNTRs to define what motifs construct VNTR alleles across populations. And then I'll get into the method for annotating VNTR composition in a bit. So what does it mean to annotate VNTR composition? So if we're going to use just this kind of little toy diagram here to illustrate how we do this. So this blue, green, and red triangles just represent different motifs that are observed at a particular VNTR sequence within a population. And we want to be able to take a new query sequence that might have tandem repeat motifs that we haven't seen yet, illustrated by this darker blue and darker green. And we want to pick a set of motifs that represents the closest sequence that we can generate to this query sequence. And we're going to call that the motif decomposition of our tandem repeat. Now, and here's kind of a motivating example. This is an intronic VNTR to the gene 7, and this was found to actually be a modifier of ALS, disease modifier of ALS. What's shown here is a large number of a large length diversity, as well as motif diversity, where each different color here represents a motif that's observed in a population that was sequenced for the study. So the problem of taking a set of input motifs and finding the closest possible sequence to a new query sequence was actually solved in the Pesner lab a couple of years ago by Tatiana Dvorinka and Andrei Vizkadze in the context of annotating centromeric sequences, where they're able to take a set of input sequences that represent monomers that construct tandem centromeres, essentially annotate what monomers are in a particular centromere. So this problem is a solved problem. And we use this approach for annotating query sequences. But what we don't have is a method to generate a well-defined set of input motifs given a population of genomes. In particular, we could take a population of genomes and essentially record all of the motifs that we see, every last one. And we can potentially take query sequences and output the exact motif decomposition where there's no difference between our decomposed sequence and the initial query sequence. But that's not necessarily going to help us in certain population studies where we don't really want to have every possible difference showing up, because that might decrease, say, the allele frequency that we're hoping to find associations between particular VNTRs and a trait or disease. What we want is what we call an efficient motif set. This is a subset of motifs from our initial set of motifs that can be used to find the decomposition of tandem repeat sequences within a specific edit distance of our original, of the set of known observed VNTR sequences, and likely within a low edit distance of new VNTR sequences that arise as queries. So what does that look like in practice? So if we look at the set of, if we take a set of assemblies and sort of count how often for one particular locus, count how often do we see every different motif across all assemblies within that locus, you tend to see a few commonly observed motifs and a bunch of rare motifs. And we could just kind of pick some frequency threshold and annotate all new motifs or all new VNTRs with the more frequently occurring VNTRs. But we elected to develop a method that essentially uses a combinatorial approach to optimize on VNTRs that, sorry, motifs that are selected from this full list of motifs that don't just optimize on frequency, but optimize on the edit distance between the full motif set and the efficient motif set that's chosen. So I won't get into the optimization method, but the general overview is that we have the initial set of motifs and their counts. We have a cost function to replace one observed motif with a different motif. And then we have an optimization step that selects this subset of motifs such that the overall cost to use this subset to represent all possible motifs is minimal. Okay, so we'll start to look at what this looks like in terms of application to a larger set of genomes. So this is taking 64 haplotype resolved assemblies and observing the total number of motifs that can be found across a database of about 420,000 VNTR sequences in the human genome. And I'm going to show a few different plots that show different values of this parameter Q. And Q is essentially a parameter that can be used to tune the amount of edit distance that we allow between the efficient motif set and the original sequences. And so if Q is 0, we allow, that's kind of the most lenient threshold. We essentially allow the full original set to be included. And the larger the Q value, the sort of greater the penalty to, greater the penalty to keeping some of the original motifs, essentially the smaller the motif set size. And so what you can see here is that even when we essentially include all motifs at 64 genomes, we're actually starting to kind of plateau in terms of the number of different motifs that we see. And this is actually fairly promising. This means that while these sequences show a very large amount of heterogeneity in terms of both length and composition, at least in terms of composition, once we get to a few hundred genomes, we'll likely have a pretty decent representation of the global diversity of VNTR motifs. One of the effects of finding this efficient motif set is essentially compressing the set of motifs that were found before. Essentially, you can think of taking this long list of motifs that some appear infrequently, and we're going to largely be replacing those with similar motifs that appear more frequently. And so the number of different motifs that you have in the full data set are going to decrease with that operation. What we can show is that there's about a 20 to 30% decrease in total efficient motif set size as we increase this sort of compression parameter. Okay, so the method that we have is called VAMOS to do this, and it has two components. There's this discovery component, which its results essentially are the last few slides that I've shown, where we take this list of coordinates of tandem repeat sequences, we take a set of input assemblies, we filter out a set of input assemblies, and as well as the tandem repeat annotations on those assemblies. We filter out sort of conflicting tandem repeat annotations, as well as, say, cyclical rotations of different motifs. And what we're left with is what we call an original motif set. And this is essentially motifs that, while they have gone under some filtration, they represent what we can use to find this effectively exact representations of tandem repeat sequences in this discovery set of genomes. And then we run this optimization step to select an efficient set, and we're left with finally a piece of data that's essentially a bed file that's a set of VNTR coordinates, and for each tandem repeat in the human genome, a list of the various motifs that we've found to be the best ones to represent that tandem repeat with. And then for sort of the future, well, the version of the software that's able to annotate new genomes, that takes as input essentially three different types of data. You can either give it haplotype resolved assemblies, which go through whole genome alignment, and then we annotate tandem repeat sequences per locus, or you can give it unaligned reads. So these are either phased aligned BAMs or aligned BAMs that aren't phased. We have kind of a quick heuristic for max cut that we use for phasing reads. And then similar to the assembly approach, we extract sequences that overlap a VNTR. We find the consensus of those sequences, and then we run them through. Then we essentially have two haplotype resolved VNTR sequences per locus, one for each allele, and we run those through the string decomposer algorithm using the efficient motif set that we defined in this discovery phase. Okay, so what does this look like in terms of what we can find and the biology that we can find with this current data that we have? So again, I mentioned before, the current human genome structural variation and human pangenome reference consortium variant databases don't really account for multi-allelic tandem repeat sequences, at least directly. When we look at the number of alleles per locus that we find when we merge calls based on proximity and similar size, we get about 2 alleles per locus, at least if we go through the data from the last Human Genome Structural Variation Consortium, or HGSVC paper. And so what we want to do is answer the question, what is the actual allelic diversity? Or in other words, how many alleles per locus do we actually see if we look at the motif annotation as opposed to this sort of length-based merging? And we use the tool almost to annotate that. And so simply we have more alleles. So when we use a fair bit of compression, we get about 3.1 alleles per locus when we annotate this set of 400,000 loci with VAMOS. And so what's shown here are the number of alleles and the frequency of how often we see different loci with that number of alleles. And so you can interpret this as, well, there's a very large number of loci that are not terribly variable. There's either one or two different alleles observed in the population. And then there's another set of loci where at 64 assemblies, we have, in this case, we cap the plot at 40, but we have 40 different haplotypes in 64 assemblies. This is with the unfiltered motif set. And then this is with the efficient motif set. The different lines that are shown here are kind of allowing for different distances between VNTR sequences, or different amounts of differences between tandem repeat sequences. And essentially, ED0 refers to an edit distance of 0 between tandem repeat sequences. This is an edit distance in motif space, so we're matching how often you see exact representations of VNTR sequences, not single base differences between sequences. And so, so even with the efficient motif set, you still see a very large number of alleles per locus, even though we're kind of compressing some of the diversity that we see in samples. So what's interesting is this is essentially the same plot as we had before, but it's contrasting the frequency of how many different alleles we observe with the human genome structural variation assemblies, so 64 assemblies, to the human pangenome reference consortium, so 90. We see essentially the same number of, the same distribution of allelic diversity. And so what that tells us is that what we'd expect to see is that if these sequences, if we had a lot more variation to discover in these sequences, we would tend to see more sequences, a higher number of sequences that have a large number of alleles. And we can kind of focus on the plot on the right, where the red line and the green line are the same edit distances between HGSVC and HPRC, and they're basically on top of each other. And so with some minor kind of bumps in the HPRC, so boosting the number of genomes by about 26 genomes that are a little bit higher quality than the HGSVC, we don't have a huge boost in diversity that we see. So that tells us that we're going to top out relatively soon with this approach. So this is just kind of a view of the VAMOS annotations of two of the loci that we've mentioned earlier in this talk. So on the ACAN, tandem repeat, and the WDR7. And so what these are illustrations to show how much diversity there is at these different compression levels. And you can sort of see where when we compress a little bit, we start to see this increased purity of some of the core domains of this tandem repeat. And what we think is that we're actually getting rid of some consensus errors in the assemblies, where this should be potentially kind of a long conserved core domain of an exactly repeated sequence, but there's slight differences when we use the initial set of motif sequences. And when we have this efficient set of motif sequences, we see what we think is a better estimate of the allele frequency of tandem repeat sequences here. But we can also see that as we get to this higher compression, that we can really see the sort of more conserved parts drop out, and we can see which regions are more similar to each other. And the WDR7 VNTR, which is much more highly variable in terms of motif composition than ACN, we can see that as we increase this compression, we're kind of left with these sort of three different motifs. There's this two sort of iterating motif, two different sort of flipping motifs on the internal part of this, and then one sort of flanking motif on the external part. We can also see this extreme diversity of motif sequences when we only use a light level of compression here. And so one question that we often get is whether or not we're able to essentially annotate with all levels of compression. So that's not currently supported in the software, but it's relatively straightforward to run it a few different times and essentially have one VCF that represents all of the different levels of tandem repeat compression. It's not too hard to do, but we don't quite have it yet. And one sort of closing item to talk about is we have it, like I said, the pipeline runs on either haplotype resolved assemblies. If you have high enough quality data, that's always the best way to go, in my opinion, because you don't have any real questions on whether or not you're mixing different haplotypes when you're trying to call a variant. But since we've also implemented this for raw reads that might be in, say, too low of coverage to have a decent assembly, we evaluated the overall accuracy. Again, this is edit distance in terms of motif assignment, not in terms of base edit distance. But we can get around 95% accuracy with a pretty low edit distance with 10-fold coverage. This is on a genome-wide analysis comparing VNTR annotations with assembly-based ground truth. And we have just a couple of percent of sequences that were actually just missing annotation based on lack of reads that overlap the sequence entirely. What you can see is once you get to about 20-fold coverage, you can get about as many regions called as 10-fold coverage. Sorry, 30-fold coverage. Okay, so that is it for the talk, and I'm happy to take questions. And again, I want to thank Tony Liu, Bita Gu, and Jingwen Ren